let's consider the potential outcomes of damage to the chromosomes by radiation. So the first thing we'll look at is restitution. Because the broken ends will basically attach to the nearest broken ends they find, there's a really good chance that the chromosome will actually be able to heal itself. This is called restitution and occurs around 95% of the time. Alternatively, though, it could attach to another broken chromosome. And this is where you get acentric and dicentric fragments. So a break in the arm of one chromosome, which then replicates, right? So it goes on to produce another identical version of itself that's also broken, will result in 4 chromosome fragments, 2 with centromeres and 2 without centromeres. Recall a centromere is basically what links a pair of sister chromatids together during the cell division. Now, when they join, the 2 chromatids with centromeres may join to form what's called a dicentric chromatid. And the 2 chromatids without centromeres may join to form what is called an acentric chromatid. Now, similarly, a break in the arms of 2 entirely different chromosomes will also result in 4 chromosome fragments, 2 with centromeres and 2 without centromeres. And again, when they form, they could potentially result in dicentric or acentric chromatids. In terms of how the broken ends are finding each other inside the cell. Now, what are the fates of these dicentric and acentric fragments? Well, the dicentric and acentric parts are going to be part of the cell division. And the acentric chromatid will not attach to the mitotic spindle because of the absence of a centromere. And such aberrations, therefore, will not be transmitted to the daughter cell. However, the dicentric fragment can attach to the mitotic spindle. Both of the 2 centromeres will orient the chromatid toward opposite poles of the cell. As the centromere migrates to opposite poles and the chromatid is basically pulled apart, it won't necessarily break in the same point at which it was originally fused together. And so this breakage will again result in a loss of genetic information and likely the cell death. Some of the curious things that can happen involve ring chromosomes. So due to the stochastic nature of the radiation, it's possible that each arm of the same chromosome can become broken. And now there are essentially 3 fragments. 2 fragments only have one end broken, but 1 fragment has both arms broken and the centromere. If these 2 broken ends connect together, then the chromosome can form a ring. This ring chromosome contains a centromere and can be replicated during DNA synthesis. However, one daughter cell will have 1 too many chromosomes and 1 daughter cell will be deficient in a chromosome. And so both daughter cells will be seriously affected, most likely resulting in the cell death. More insidious and discrete chromosome changes can occur, which are not immediately lethal, but can be damaging to the cell. So translocations is when two breaks occur in different chromosomes, and the broken parts are switched between the two different chromosomes. This is not only seen during irradiation, but can also be a part of the disease process itself. For example, this is often found in leukemia patients even prior to irradiation. Deletions and inversions can also occur. This is when two breaks occur on the same arm of the same chromosome. The piece in the middle, with two broken ends, can then be deleted or flipped around and the DNA material inverted. This changes the sequence of the genes on the chromosome. In somatic cells, the necessary number of chromosomes is still present, and so while there might not be any immediately lethal consequences to the cell, the function and interrelationships between these genes could be altered. Let's look at the frequency at which these aberrations, these alterations to the DNA and the chromosomes, can occur as a function of dose. The figure shows the relationship between dose and the mean fraction of cells with aberrations, mainly translocations, measured by conventional staining in survivors exposed in the typical Japanese houses after Hiroshima and Nagasaki. The small but consistent differences between the two cities may be due to different estimates in the dose. So, as I recall, it has been argued that the houses in Nagasaki were more sturdy. They're more likely to have concrete in them, and that might have accounted for a different estimate in terms of the shielding effects. There may also have been different ways of counting the aberrations between the two laboratories that did the different studies, but clearly there's a correlation, right? The more dose that the people were subjected to, the more aberrations that were found within those cells. Now, in terms of the low doses, then, chromosomes breaks result from the passage of a single charged particle, and the consequence of this is a linear function of dose. Okay, so this is one particle that's coming in, and it is breaking both of the, both of those two chromosomes at the same time, right? It's sort of causing a damage to or in other ways to put it, is like, the DNA is the two strands. It's causing damage to both of those strands at the same time. Alternatively, at higher doses, the chromosome break could be caused by the passage of two different particles, which is basically one particle comes in and breaks one strand, and then another particle comes in and breaks another strand. And this causes a quadratic function of dose. I just realized that the picture here on this, and I forget which book I got this from, but the picture on this slide is actually showing different chromosomes being broken by radiation. You got to remember, to break the chromosome, you generally need to have double strand breaks. So that could be because of one particle, but it could be because of two. So the linear quadratic, which is the classic model for this, is usually referred to in terms of the strand breakage, not individual chromosomes. But we'll get back to that later. Recall that the curve for high LET is more linear though, because the high LET, with those big bowling balls of radiation, they cause the damage directly, and a single particle of radiation is usually sufficient to cause the damage, and so the curve is more linear because one particle is coming in and causing all that damage. Whereas for low LET, you're more likely to require two separate particles to come in and cause those chromosome breaks. So the aberrations increase quadratically with the dose, right? The probability is proportional to dose squared, and we'll talk about this later in the course. But the reason why it's dose squared, it's almost like if you toss a coin and it comes up tails, then there's a 50-50 chance. But if you were to toss the coin again, then there's another 50-50 chance of it coming up tails. But to find out the probability of doing tails twice in a row, you have to square it, right? So you end up doing 50% or a half squared. So you end up with a quarter. So then there's a 25% chance of you flipping a tails and then flipping another tails, right? So that's why it varies with dose squared. But we'll come back to that later in the course, that is. The last thing I want to talk about, though, from this chapter is mutations. And recall that any change in genetic information are termed mutations, or defined as being mutations. Now, while some mutations will result in the death of the cell, all mutations can have important implications. So, for example, the mutation of somatic cells not involved in reproduction, can have important consequences for that individual. But germ cells, if they become mutated, they can affect the general population, because the germ cells are the ones that carry the mutation to future generations. And that's one of the concerns about this type of radiation damage. Is it going to be passed on to the offspring? Well, that finishes the videos for this chapter.